Hello everyone, let us answer this question from a previous Life Sciences grade 11 question paper. This particular question is from the topic microorganisms. So 3.1, bacteria are prokaryotes. The diagram below shows a bacterial cell. The first question, state what is meant by the term prokaryote. Remember guys, we have prokaryotic organisms and eukaryotic organisms. Prokaryotic organisms, which is basically prokaryotes, these are organisms without a true nucleus. Then eukaryotic organisms, these are organisms with a true nucleus in their cell. So prokaryote organisms without a true nucleus in their cell or cells. Okay, cell or cells. Obviously, it will be one cell for a bacteria. All right, or you could have said organisms without a definite nucleus. You could have also spoken about the membranes. It doesn't have membranes in their structures that we find within the cells. So give labels for 2 and 3. So there is 2, structure labeled 2, and there is structure labeled 3. So 2 is this middle layer here, which is the cell wall. Remember, on the outside, you'll have the slime capsule, then the cell wall, then the cell membrane. So 2 is the cell wall. Then 3 is the nucleoid, which will contain the DNA of this bacterial cell. 3.1.3, give the function of the part labeled 1. So this is the part labeled 1, which is the slime capsule. So let's give the function is to protect the cell from drying out, desiccation, or you can even say to protect it from antibiotics. But I would say the main correct one is to protect the cell from drying out. The structure labeled 4, there is the structure labeled 4 is a circular DNA called a plasmid. Describe, for 5 marks guys, describe how it can be used in the manufacture of insulin for diabetics. You need to describe how the structure labeled 4, which is plasmid, how it can be used in the manufacture of insulin for diabetes. 5 marks. Let me see if I'll have space. Oh, but then I still have a 3-mark question. So I'm going to write the answer here, guys. Okay. Here's the answer, guys. Plasmid DNA from the bacterial cell will be removed and cut with enzymes. Then also the human DNA with the insulin gene is also cut and inserted into the cut plasmid. The plasmid is then reinserted to the bacterial cell to produce human insulin. Then the human insulin is isolated and used to treat diabetics. That's the answer. Develop resistance. How bacteria develop resistance to antibiotics and how we as humans can contribute to this particular phenomenon. Okay, what's happening now? All right, so the answer, so the first 2 points are answering the first part of the question. So mutation may occur spontaneously in bacteria, and some mutation may cause the bacteria to be resistant to antibiotics. And the second part is answering, or this point is answering the second part of the question about how humans can contribute to this, how can contribute to the bacteria becoming resistant. So that's obviously by stopping to take antibiotics early in the course. So if you take the antibiotics, then you stop before completing the course, which can be 6 or 7 or 8 or 9 months. All right.